Welcome back, everybody, to our third presentation of the day. This morning, Suzanne Lang talked to us about Devi Mahatmyam, goddess worship. And this afternoon, earlier, Manorama brought to us the Bhagavad Gita. And now I'm going to have Indra introduce Sarah and Paras for their presentation on brain.
Bodhonya sadhanebhyo hi Bodhonya sadhanebhyo hi Sakshan mokshaika sadhanam Sakshan mokshaika sadhanam. So, we are going to have a wonderful example of the path of Jnana Yoga here today. And that is Shankaracharya's perspective on Jnana Yoga from many years ago. So it's my privilege to introduce Paras and Sarah, Ken and Dr. Mulukutla. We know them both by many names. For me, the most emotional part is that Ken came here as a monk, as a Jain monk, in the early mid-70s. And at that time, he spent 7 years studying profound Jain texts and being very much encouraged in the study of the brain by his teachers. So he took that encouragement and got a doctorate in brain-related items and is currently the head of the Haskins Institute at Yale, where he continues to study brains. And without having lost the perspective on why do we study and what is our path, which is that of jnana and understanding self-realization. And Dr. Mulukutla is his wonderful colleague, also graduate and now faculty member at Yale, a neurologist whose specialty is Parkinson's. And she is the two of them together are an incredible source of knowledge about everything, not just to do with the brains, but specifically what happens to brains when we meditate, and what is the power of meditation to make changes in our brains, and many more things about which they will talk.
Well, Ken is going to lead for the most part. I just wanted to introduce because I'm a little bit of a sidekick today, but I'll be demonstrating the brain in 3D. I'll be moderating questions and jumping in as needed. We just wanted to explain a little bit about the format. But what Ken is going to show today is very exciting analysis that was conducted last year at Ananda Ashram. He's going to preface it with a little bit of context. But once we get into the analysis, this is what he and his lab have been furiously working on for the past few weeks in preparation for today. So like all of you, I'm still learning a bit, so we're all going to jump in and can't wait to see what Ken is really going to show us about what the study showed. Take it away, Ken. You have your study.
Thank you, Sarah. Let me turn this.
And yet not in front of the screen.
Just maybe a little lower. Let me just begin with a mantra. Om Namo Arihantanam Namo Siddhanam Namo Yariyanam Namo Vajayanam Namo Loe Savasahunam Yeshobhanchanamakaru Savabhavapanasano Mangalanam Che Savesim Paramam Havai Mangalam Om Hrim Shrim Klim Shri Parshanataya Namah. This presentation today is about a work in progress still, but where we finally have a first look at data from a study, as Sarah mentioned, that we ran here a year ago during this, well, actually I think it might have been during this, the earlier meeting we had. I can't remember if it was this one or, I think it was the earlier, not 2 years ago, it was the June meeting before we came back for the Sanskrit. So it was about a little over a year ago. And this comes out of a long conversation and set of friendships with Shanta and Indira and Sarah and myself, Hari and others, who have been wanting to do more with bringing some of the tools of neuroscience to both inform and to learn from the deep practices that go on in an ashram. Dr. Mishra had, of course, a vision of the East-West Sanskrit, East-West music, East-West dance, and East-West Jnana Yoga, or knowledge. And this was really a team effort. We thought, wouldn't it be nice if we could bring the Sanskrit meetings and the study of the brain and language and meditation together somehow, and sort of create a dialogue. And I think that's exactly what has happened. We've had a number of interesting discussions while we were waiting for this data, which took a long time to cook up. And Sarah and I have been working together, and she can speak about projects that look at in her own work, look at Parkinson's patients using these kinds of brain imaging and the impact of mindfulness training, which Sarah has done extraordinary work to really benefit patients with severe symptomology. And then we've been extending this in our collaboration at Yale now to other populations and other projects, including my own work with children who struggle with attention and language and other issues, where we're now setting up to run some of these measures as part of the preparing children for learning to deal with anxiety, which is really very much off the scale these days in young people, and really a problem, and for which mindful meditation seems to directly impact the same brain pathways, the same parts of the nervous system that are impacted in anxiety. So a lot has come out of this conversations we had. But in order to, and Seth, can I? So just as Sarah said, I want to give a little background, because the experiment we ran here was really the culmination of a lot of conversations about should we study language, should we study chanting, should we study receptive Sanskrit, what should we do with Devanagari and the writing system that needs to get situated in your brain as you become somebody able to become biliterate, or able to read two languages, as well as bilingual to speak them. So we had a lot of wonderful conversations and a lot of planning. And that led to the study that we were able to run upstairs in the music room. First of all, there are a number of people, and I don't want to forget this at the end, that I want to thank for bringing this study to the ashram. And so I begin with two of my research associates, Gladys Venegas and Virginia Chambers. Some of you may remember they were here putting caps on people all weekend and got 5 minutes to swim. But they were amazing in actually collecting brain data in the field in the ashram. And then two colleagues of mine, other than, of course, Sarah and I, two colleagues of mine who've been really diving deep into this preliminary study, Shen Zhang from the Department of Psychiatry at Yale. He's a mathematician and imaging analyst. We worked together on a number of grants. And my postdoc, Ahana Bajracharya, who's originally from Nepal, is an engineer, neuroscientist, did her PhD at Washington University in imaging science and engineering, and she's extraordinary, has helped us to get this data up and running. So let me remember, I don't want to forget that at the end, to thank the people that have been working hard to make the presentation. So a very quick look through some background, the background that went into us deciding to do this study together. First of all, as Indira chanted, the path of Jnana Yoga is attractive to some and to me. And we know that Jnana Yoga aims to know how things work and our place in the universe. And the idea is that the better the understanding, the better the information, the better the spiritual progress. That somehow science and spiritual matters can mutually inform each other. And to be honest, I have this extraordinary technology at our hands to look at the very tiny parts of the brain, the cellular level or the systems level that you'll see in a few minutes. And I know that the ancient yogis, the Gyanis, would have just gone crazy. And all of the discoveries on genetics and how they build the brain is very much reflecting some of the ancient speculation in the Jains on karma as memory and particle, and a sort of an organizing material process that creates habit and bodies and life and experience and capacities. So the Jnana path, I think, benefits from modern science by having so much lens into the biology of the Prakriti, if you will.
Just keeping you on track, we have 45 minutes before we have to
Okay. So, and then why study the brain? Because this is a biological machine that we need to conquer and control in our practices, as we know. And this is what I tell my students. I think the Jnani neuroscience dialogue says the going deep into our practices, to go beyond the mind, is a steep mountain. And an understanding of the brain and its vulnerabilities and its strengths, in my view, and I think Sarah probably shares this, it can become a roadmap to help us move through and gravitate to the deeper parts of the brain. So thank you for the reminder, Sarah. So let me see if this is working. Good. So we used a kind of neuroimaging you'll see in a few minutes called functional near-infrared spectroscopy. Let me just give you the briefest overview of what we mean by brain imaging. So this is a brain, this is a person, and in brain imaging research, we use a variety of tools like functional magnetic resonance imaging and EEG, and the FNIRS, which I'll show you in a minute, that we used here upstairs at the ashram. And we acquired data on where and how knowledge and processing, things like remembering, speaking, listening, deciding, creating, solving problems, all of this is taking place in this complex array of systems, and these give us a reasonable view. We can also look at the impact of the change in brain states and consciousness, awake, sleeping, coma, lesion, brain injury, and how that changes the circuits. And we're able to even stimulate the brain. Some of you may have heard the work these days with deep stimulation on certain brain areas that have been somewhat helpful for some who struggle with intractable depression. So we have a variety of tools. Very simply, one of the ways we measure the brain, and the way we're measuring it with the FNIRS here that I'm going to show you, is to look at areas of the brain that change their blood flow based on activity. So let me give you the logic. You are in this experiment and you're doing one of two things. You're either opening your eyes or you're closing your eyes. So when you open your eyes, the visual system is getting activated. When you close them, it's not. And so you can identify changes in what's called the blood oxygen level dependent signal. Essentially, in English, it means that when a population of neurons becomes very active, a few seconds later, there's a profusion of oxygenated hemoglobin in the brain, and that has a different magnetic signal, a different signal than deoxyhemoglobin. And so, in a nutshell, we're able to use this to identify where things are happening in the brain. And you can see here that in the parts of the brain in the visual cortex. You can see that there are regions that are increasing when the eyes are open because you're seeing, and decreasing. So this gives you just that feeling that we can localize things in the brain based on this kind of information.
And just to help people just orient
Oh, yes.
what this is, just to make it easier, but this is sort of what a 3D brain looks like. It has two hemispheres. This is challenging with a microphone, but what he was showing you was a side image, which you'll see a lot. Excuse me. Including what's here, and the vision is in the back of the brain. So that's the way that it was oriented. That was the back of the brain, and that's just a well-known area where the brain is.
Yeah, so, and you can see that showing up. So look, we're able to localize systems in the brain. We also measure, and this is important for where we're going to be going next with this study, so I'll just mention it briefly. We also can use EEG, which measures electrical activity on the scalp. Sarah, as a neurologist, would be using this quite a bit. And this gives us information about the timing of brain activity when people are doing different things like listening, speaking, remembering, singing, etc. And we can identify both the location here and the timing of these things. And by combining these measures, you get a very good characterization of how the brain is operating when you're doing something like chanting Sanskrit or meditating or anything of the sort. And the last thing I want to show you, just briefly, this is the tool that we brought upstairs. It's called near infrared spectroscopy, and I'm not going to dive into nerdland. Let me explain what it does. There is a kind of an emitter and a detector. Does anyone remember we had the caps, cute little caps, and we had the little light going? So what happens is there is a signal, an infrared light, that passes through the skull and enters the cortex, the surface of the cortex, where a lot of cognition and remembering and perceiving takes place. And then it bounces back, and it's detected by the detector. And in this way, we can measure where there are changes in that oxygen signal. The MRI machine is big and bulky and complicated. This machine, this device, as you might have noticed, is portable. We can take it anywhere. And we get a lot of useful information. So this is the thing I want you to remember. What do we look for when we study the brain activity? Well, we can always say, well, where is it? You know, when you think about truth or beauty, where is that in the brain? Well, it's not in one place, obviously. So what we look for in our research...
You're just in the middle of the slide, yeah. Good.
Yeah, so what we look for in our research is areas of the brain that are operating as a network or as a set of hubs and nodes, right? When you are processing information, it's not one part that does anything. One part talks with other parts, and they combine their activation, they combine the processing, and we are very much interested in almost all areas of science now, including the studies of meditation, in not where specific parts are, but how they're operating as a circuit or a system. And we refer to this as functional connectivity. Go ahead, Sarah.
No, I will add that meditation was one of the first studies that showed that you can very quickly reorganize the functional circuits in the brain. Meditation is one of the quickest tasks. They definitively showed that the meditation of all kinds, but a specific one that was studied, changes those network connectivity within a few weeks.
And you can see here, what Sarah's alluding to is that one network in the brain that is associated with attention shifting and even what we call time traveling, like remembering or predicting, we call this the default mode network, and other networks are more active when you have to focus on one thing and ignore other things. So there's a lot of these circuits, these networks. And when you're meditating, as Sarah is saying, you can see the mind wandering system, which is a horrible misnomer, but the mind wandering system tends to reduce as the mind becomes more focused. So all this is telling us where and how things look in the brain. And I've got to share my favorite slide. So there's a group in UCSF, Adam Ghazali, and what they did is they took 3 kinds of brain data. I didn't give you all the boring details. One measures the gray matter structures. It's called structural MRI. And it tells us where the different folds and gyruses are in the brain. The second one is a thing that measures the axonal tracts that connect neurons in one part of the brain to other parts so that they can work together. These are long axons. And with this technique, you're able to get a very clear map of one person's brain. And now, using some high D graphics, they remove the gray so you can see. This is a real data. This is a real subject. I went back and looked it up. Jayshree was asking me last time, what were they doing? They had them open or close their eyes and squeeze a bulb. So it was really just a boring thing. But watch what's happening. Because the data that is giving you those little flashing lights, that's where activity is while people are cognitive using the EEG. So this uses different imaging to give you, and because it's high tech, they're going to dive in and show you all of those pathways that are active when you just do something as simple as seeing and not seeing. So the brain is extraordinary, and we'll leave it at that. The second thing is language is also extraordinary. And so we spent a lot of time thinking about, well, the brain's cool, and there's a lot of lessons, there's a lot of potential value added to a yogi, in my mind. And there's a great lesson that yoga and deep meditation provide to modern medicine and modern science, even basic neuroscience. The study of plasticity, the ability of the brain to reorganize itself, has been greatly enhanced by the study of intensive meditation training, because you're basically changing all the connections. So it's a very exciting time, and there's a bi-directional communication. Language and Sanskrit are always interesting. So I'm just going to show you that there is a great deal of research from our lab and many other labs around the world on where and how language nodes in those networks tend to look in the brain. So if you look at this one, just briefly, there are areas in the temporal lobe, and Sarah can point to these.
It's kind of right in this groove. It's on the top part of this groove over here.
And these are very critical for receptive language, understanding what I'm saying and not mixing it up, so the expressive side.
Yeah.
There are other areas that are involved more in articulation or output, which are shown here, the so-called Broca's area.
Can I take a pause for just 1 second?
Yeah, please.
I just, what I'm seeing here, the different areas they're showing are the many different aspects of language. So one is, it's called perception, right? So if you're listening to me, part of your brain is understanding. And that's what they're trying to show there. And as I talk, I have to make the sounds, my lips have to make the sound, and it's all coordinated and how quickly it happens in the brain. But even though you're listening and talking at the same time, these different areas of the brain are on and off at the same time. Right. And then those pink is just that's the whole understanding of language? Is that what they're saying? That's the comprehension of language?
So basically this is a summary of a lot of their studies, and as Sarah's saying, you have areas in the posterior, in the temporal lobe, that are critical for receptive understanding, and then areas in the motor output that are critical for speaking. So there's a speaking-listening dynamic here, but then what do you do with that? Well, you pass it on to understand what somebody's saying, to understand the message, to understand that if I say it felt like a rainy day, I'm not talking about rain, I'm talking about my mood. So all of this is elaborate and involves a wide array of brain areas. And what are they doing? They look like that rotating brain. They're going jing, 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 and they're communicating and they're talking to one another unless there's brain injury or if there is some disorder of the brain. So this is just to give you that feeling. And one last thing before we switch to our data. And it's really intriguing. right now. So when we study the brain, reading, listening, remembering, thinking, being conscious versus being asleep, we are able to see all these areas and try to make sense of individual differences and why some people struggle and some don't. But when I'm looking at, Girija's brain, I tend to see it as there and Ken's brain here. And one of the things that is emerging in sort of the periphery of creativity in neuroscience right now is this understanding, as shown in this paper by Uri Hassan's group at Princeton, that when two people are communicating, there's actually an entrainment that occurs. So the patterns of brain activity in the perceiver, to the degree they understand the message of the speaker, the story, the patterns you see over 15 minutes of brain activity are highly correlated. So what this means is you're not going to really understand how the brain communicates or meditates or thinks if you're only looking at a brain in a vacuum, because we are in fact brains that live in context. Jayshree and I always say this, there's something about meditating in a group that you can really feel. And I just think it's fascinating. And the reason I threw it in, Sarah's going to yell, but the reason I'm throwing it in right now is I just want you to understand that the science is progressing to a point where we're thinking about the social brain. In fact, there are studies that are going on where everybody's got a cap. You got a cap, you got a cap, you got a cap, whatever, like Oprah. But we're basically looking at how good communication is associated with this kind of entrainment. So, mindfulness and brain, as Sarah already and I have mentioned, there is a fair amount of research showing that when you go deeper into different states of meditation or you vary in your expertise, learn, experience it, go from a focal meditation like mantra to witnessing to contemplation, or what the Jains would call bhavanas, so do the Buddhists. These are have different brain pathways or networks that are involved. So now we come to why we're here today. So that was background. And why did I give it to you? One, Jnana Yogi's cool. Jnana Yoga's cool, maybe. Two, the brain's interesting because to understand the map might help us in ways that you'd be surprised. In fact, my last slide, if we get to it, is one that shows how with people that have difficulties in meditating, if you understand the causal networks, you can actually facilitate the meditation itself. There's some new research, and it needs to be replicated, but it's very interesting. So, neuroscience and meditation are there. Language, this is the first time, Indira, that I've spent such a short amount of time on language, because I'm a language guy. But I just want to remind you, there's expressive, receptive, comprehension. There's also entrainment and communication. These are things that need to be understood. So when we were sitting around the ashram...
Wait, do you want to take a pause? Does anybody have any
any questions? Because now we're switching to the data.
And by the way, you can say your question, but we're going to repeat it. So
yes.
Please, yes.
Hari.
My question is real quick. Were you able to get any data on meditators who are trained or who are experienced doing it for many years, who know how to do it,
Yeah.
versus new people who struggle to meditate? Did you see any sort of fMR that shows like this brain is actually constantly active and thinking versus a calm brain?
Yeah.
You're
So, go ahead, Sarah.
No, you're talking about in general, right? Are there any studies that show?
Right.
Or did you find anything
Well,
else
we haven't gotten to our study yet.
Well,
but
the problem with us is that we have a lot of very experienced meditators and only a couple of novices. We were only able to run and get usable data on 8 people, 8 subjects. But I'm going to show it to you and show you what we've learned. The literature, though, Hari, is clear that there are a variety of biomarkers, subtle and connectivity-based, that are associated with depth of experience, number of years, quality of meditation, type of meditation. So we have a lot of things we are hypothesized. This data, just let me get to it and show you, is I have to make a big decision, and the decision is, do we have enough good data here in this design that we constructed together to run more people? And my answer is going to be yes. Yeah, Seth and Laraji.
I just want to piggyback on Hari's question.
Yeah.
And you just alluded to the sample size, which I believe you said is 8.
On this one, yeah.
Yeah, so obviously a sample size of 8 is very limited, as opposed to going to Times Square and selecting a random 1,000 people. So there's that issue. But also, what about the issue of the fact that we collected it at an ashram, where you're kind of self-selecting for people that are...
Oh, yeah, no, absolutely.
Let's repeat the question for the anyone who's
Yeah.
on live stream. So Seth's question just now was, aside from the sample size, can we also address the fact that we collected this data at an ashram, which is different than any other setting or specifically a lab? The question before had to do with, was there any differences between novice and experienced meditators? And that's where Ken said that the literature that is published, yes, shows a lot of difference in, we can well characterize how the brain changes, becomes plastic through the act of meditation itself. Okay, to Seth's question.
Exactly. Those are the two answers. And Laraji, you were going to ask?
Yes, does your examination in this area arise any curiosity about an abdominal brain?
Yes.
The last question was does this examination arise any curiosity about an abdominal brain?
Dr. Mulukutla.
Answer Seth's question, and then Laraji, and then we're going to move on because we have about
Okay.
15 minutes to
Yeah.
review our data.
No,
so
we're
we'll live it.
scheduled until 5.
We want to finish 4:45 for discussion, so that's our aim. Okay?
Well, it's only 4:20.
21.
Okay. So, and we also started 5 minutes later than, so at 5 o'clock I think was our end date. I don't want to be that rushed. But please, in terms of gut brain, do you want to say anything on that?
Oh, there are neurons in the brain. The gut has sensitivity for sure. I actually didn't realize it was a true question, but I'm happy to answer it. That sure, yeah.
Well, I mean, basically.
Yeah. The gut and the brain are intimately connected. There's many neurons. The gut produces a lot of the neurotransmitters the brain uses. It's the largest producer of serotonin in the body. The abdomen. Yeah, they're connected. The whole body is connected, actually. I mean, you can ask for a cellular brain because they have receptors. If you talk about a brain as a sensing and active organ, then all of life everywhere is a brain. But we're talking about the discrete organ in the skull.
But and of course there's this growing research. There's much known about things like the anxiety or stress response, the fight-flight response, and how it depends on a variety of known pathways, the hypothalamus, the pituitary, the adrenals. But there's a growing evidence on what's called the gut brain. And this complex array, this lattice work of neurons that follow the intestinal system, there's at least a fair amount of recent publications that this brain, this extension into the gut is impacting a variety of behaviors. It's impacting mood, it's impacting anxiety, and it's really a hot topic right now. So I don't know beyond that what to say, except that at least in the fields that I'm working, people are taking a growing interest in how this contributes to health and to physical health and mental health, and it's an area that's exciting in many ways.
I haven't heard of EEG yet for the gut, but maybe that's coming soon.
Well, you could certainly do MRI.
Yeah.
You could look at, although these are pretty small. But yeah, go ahead, and then go ahead, John and Hari.
Quickly, I read someplace that in ancient times, people, if you asked them to point at themselves, they would point to themselves.
Yeah, of course. Yeah.
Okay. The comment for the audience is that if in ancient times when people would be asked to point to themselves, many would point to the gut. I thought it was the heart. The heart
even now.
more so. The heart or the gut? Yeah. Even the gut? Okay. In particular, in the belly.
Where you sense from.
Exactly. No, this is exactly, and it is. Go ahead, Laraji.
No, I was just seeing how that felt. Me.
Yeah.
Me.
Yeah. Yeah. No, this is, first of all, there's a big area in my field that's called embodied cognition. The topic, Laraji, is to understand how we learn through our bodies. This is like another lecture, and I won't go here right now. But embodied cognition is the sense that we organize our understanding of the world and our language around motor systems, around action, and around the body. And it's fascinating to see how that shapes language and how it shapes experience. The fact that we are not just these disembodied brains, but these motor. In fact, there's even a theory of consciousness that Hannah and Tony Damasio, two neurologists from UCLA, put out on how consciousness itself, in part, can arise from the feedback we get from our gut and from our biology. So it's a fascinating area, but let me not go further because I want to show the data.
But I want to ask you.
Yeah, Hari.
I think the real question I wanted to ask got lost. If you take a sample of 100 people and you did an fMR analysis and obtained the data,
Right.
can you say conceivably, using your methodology, 50 people have been meditating for 10 years, but doing it the wrong way, meaning they're not making any progress, versus 50 that somehow know how to meditate and they're successful. Can you tell them?
Well, so that.
So tell the people that don't know how to meditate and struggling,
Yeah.
give them information how to help them.
So, go ahead, Sarah.
Go ahead.
Well, I was just going to say, so this use
Repeat
of.
the question.
So Hari is asking if you were to use brain imaging and you were to look at the patterns, these complex network patterns, in 100 people, 50 of whom were practicing 10 years but not doing it right, and 50 of whom are getting success, would you be able to, the way we do it in statistics is we say, could you look at the scan and put it in the right bin? Okay, this kind of discriminant group analysis. And I don't know of any studies on that, but I know there's enough consistency on what deep meditation tends to look like relative to struggling and anxious minds that I would imagine you would be well above chance, doing the study, of putting people in the right bin. How that would go to what to do about it is the fascinating question. That's where the neuroscience is interesting, because if we know where they're off track, we might, if it's in the affect system, if it's in the attentional control systems, we might have ideas, we might have ways of recommending. But I would just say I'd be open to that.
Well,
Go ahead, Sarah.
There's biofeedback systems now,
Yes.
where you put a headband on
Ah,
it,
of
and it
course.
reads your activity in real time. And then actually they make you control a video game, and they make you, when you get it to go, then you're activating the part of the brain that they want to activate.
Exactly.
But it all comes down to finding the biomarkers. You have to know what you're looking at to then, first you have to know you have to do a lot of research to figure out what the effect is physiologically, and then you'd have to map it onto probably each individual brain. So it's complex, but it's...
And that's where the neurofeedback is one area that's rapidly growing. And another one that I mentioned earlier is because you can look at the brain areas that are involved in meditation, and then you can use this thing called transcranial magnetic stimulation, which stimulates. And they're just starting, and this was the Pascal Leon group up at MIT that did really some very good work on showing it's kind of like which areas are causal and which ones are just active. But that is where meditation as a tool for mental health, because people who struggle often can't meditate, that's where the neuroscience is low-hanging fruit. It's going to help. For somebody who's going for enlightenment, then the deeper question is, what do we need and what does science tell us? I'm a Jnani. I think it tells me a lot, but we'll continue. Let me show you the data, because that's why we're here. So we designed this study, and we brought a cap that measures this hemodynamic signal, and this was a very limited cap. It had 16 of these optodes, and it was measuring the left and the right hemisphere, and we placed it in the place where we thought we could get the maximum impact of meditation. And what did we do? And Indira, you'll remember this well. So what we did is we designed a study in which we had subjects put the cap on, and do something we call resting state. And I'm going to show you this in a minute. It's a way of just making the mind just relax and go into a kind of a baseline state. I'm going to show you how we do that. And we did that before the meditation for 6 minutes. Then subjects were assigned, people, ashram people, to 2 groups. Now, we don't have enough data to publish this yet, but I'm beginning to think that we might have some patterns that are worth getting more subjects. So one group listened to Indira chanting Sanskrit shlokas, and the other group listened and repeated as we do here, right? So that kind of shadowing, where she would read and then the subject recites. So one was receptive, and the other was receptive plus expressive. And we thought, Shanta, that maybe that would have a different impact on meditation, this articulation, this chanting. And we knew this wasn't going to be the study to end all and be all for everything, right? But it was like, well, we could do that. So we had 6 minutes, subjects listened, or they listened and repeated, sort of line by line. Then they sat for 20 minutes and meditated. And we gave them a questionnaire afterwards, how do you meditate? And how many years have you meditated? Unfortunately, we didn't have too many beginners. But we can get them up at, if we want to run more subjects, I can get a lot of beginners. But for 20 minutes, they simply meditated. And then at the end of that, we did this thing called Inscape, I'm going to show you, to look at what changed in your brain networks from before to after. So this is, each subject was assigned to either the listening, repeat, or listen. And unfortunately, we've got only 8. That was a really long weekend, though, and it was amazing we got 8 in one weekend. And then this is how many minutes we do. And these were the key questions in this design. First, if you look at the brain before and after chanting Sanskrit and 20-minute meditation, has it changed? That's the big question. And we use this thing called Inscape as a marker of the brain's baseline network. 2, is there a difference between listening and listening plus chanting? I need more than 4 subjects, damn it, in each group. I see there's actually some patterns in the data, but I don't trust them yet. So I'll have to get more subjects and fill you in. But that was, of course, we talked in India a lot about that. And then the meditation session itself, which is similar to what Sarah's doing with her Parkinson's patients. They do this Inscape before and after they do the meditation. And we were looking at the general pattern of activity. And also, is there a change in activity from the first 10 minutes to the last 10 minutes? What happens between the first 10 and the last 10? Usually meditation gets deeper. One hopes. Not always, but more or less. So we were looking at the effect of pre versus post on brain, the effects of listening and listening plus chanting, the effects of the first 10 minutes versus the second. And the measures we look, and this is nerdland, but it's not that complicated. Number 1, the brain activation in each of the areas we were able to measure, the signal, how strongly they were activated. The second one is functional connectivity that I was talking about and Sarah was, where we look not at the one area, but at the communication between multiple areas. And the last one, which turns out to be it's going to win the race here, is something called moment-to-moment variability, and I'll explain that when we get to it. So we're looking at a variety of hypotheses. If you meditate and do good Sanskrit, there's going to be a change in the efficiency of the baseline network from pre to post. If you listen or listen plus chant, there'll be a deeper meditation. We don't have enough subjects for that yet. If you watch the brain meditate over a 20-minute period, there are going to be reliable changes that are associated with how deeply, Hari, you're meditating. And finally, we measure it in these ways. This is what Inscape looks like. So this is what we do before and after, and I've only got a 15-minute 15-second, what do you call it?
Clip.
So listen. And watch. You're sitting there. That goes on for...
Meditation of itself.
That goes on for 6 minutes. It keeps your visual system and your music system active, but it's got nothing to do. It's just nice, pleasant. And we do that for 6 minutes before, that was only 15 seconds, and 6 minutes after. We do 6 minutes of chanting or listening, 20 minutes of meditation. So that's the basic design.
Okay, let's see if we can try and get this all.
Okay, so here we are looking now at results. So the first result I'm showing you is a measure of which areas are differentially active in those different parts. The inscape, the meditation, the chanting. And which areas are showing that pattern together even at different parts of the brain. So there were two that were very strong. One is, and this is something called independent components analysis, and this is just a way of looking at complex multivariate data. I'm not going to
No, but we can say that basically
Go ahead.
they collect the data and there's a lot of processing that has to take place because we're looking at basically blood flow and the type of blood, and so they have to remove the pulsations of the heart, they have to remove background noise, sometimes there could be like some filters, so there's a lot of processing that takes place.
Right. And what you're looking at, so for example, I'm saying...
What do the different numbers mean, though?
So
Like,
these are different components. This is a statistical...
That they're removing, basically.
Yeah, now they're basically, these are sources of shared variance in the signal that are associated with different parts and different brain regions. So this is a way of looking at connectivity and activity. Let me just show you, for example. The areas here in the left and right frontal cortex and the left motor sensory region were strongly correlated in showing a pattern where the second inscape, the after meditation, which is shown in purple, is higher than before. So something big changed in these regions in that signal. This...
And red and blue, they mean active or deactive?
No, they're positively correlated or negatively correlated. This is just the actual tasks. So what this one is saying in a nutshell is there were a number of brain regions that changed pretty strongly on average when you did the inscape before and after. So now we know what to look for. These are where they are, what are they doing, are they associated with how many years you've meditated, and lots of other questions. Are they associated with whether you chanted or you didn't chant? Damn it, I only have 4 in each group. I need more. But we'll look at that when we get more. The last one I'll show you here is another statistically significant component from this complex analysis, which shows regions that are more medial, frontal, and parietal. And these are areas that are changing the most during 20 minutes from the first 10 minutes to the last. And these are areas that are essentially becoming less active over the course of the meditation. So we would be targeting these regions to look at chanting, Sanskrit, no Sanskrit, how deep the meditation, whether you're a 50-year meditator or a 5-month meditator, and we would expect it to be affecting these different tasks.
I want to add just one thing, that just reminding everybody,
Yeah.
from this view, you're kind of looking straight from the top, and just reminding that there was only coverage for really that area. So
Right.
we don't know exactly what happened in the very front or the very back. We just didn't have the electrodes to cover that.
And this is where we're going to end, because we're going to say, I have a much better system now, so we can get better coverage. Go ahead.
Negatively correlated means less activity or less coherent.
So the activity has this pattern, but if it's red and blue in these plots, it means that these regions are negatively correlated. They're not, or positively correlated, because the brains. You saw that beautiful brain. Some areas work together and some inhibit one another, and this reveals, like it shows us, what the pattern is that is associated with positive connections in some regions and negative correlations or anti-correlations in other regions. It's complicated, but what this shows us is that, in English, is it shows us that there are reliable markers of which of these different components of the experiment you're in. Even with 8 subjects, we're able to see these patterns with statistical reliance. And that will in turn be where we look closely to ask the deeper questions with a larger sample and better brain coverage. Let me just move on to 2 last things. Oh, go ahead, Indira.
I'm really curious about the Inkscape thing.
Yeah, I'm going to get to that.
Okay.
Yeah.
I'll wait.
Yeah, the Inkscape is where the action turns out to be. This is just the general way in which we look at which brain networks are hanging together for different tasks. That's it in a nutshell. And I'm confident, I'm happy that we're able to see this much signal even though we had limited coverage and a small sample.
Were you surprised by any of these findings?
The last one I'm going to show you. So the other thing we measure is we look at each of the regions we covered, and we looked at what's called functional connectivity. That's the correlation. You have all those different regions, a lot of regions, and you're looking at the degree to which they are correlated positively, which means as one goes up, the other goes up, and if one goes down, the other, and anti-correlated, one goes up, the other goes down. And it's a complicated array, but what we were able to see is that when we look at functional connectivity during the 20 minutes, there are a variety of brain areas that are sort of negatively correlated. The brain is parcellating out in some way. I don't want to make too much of this with 8 subjects. I mean, I'm a scientist first, but I can see that, you know, some of these things made some sort of conventional statistic. If you look at the positive correlations, we see, for example, that areas in the frontal and the temporal lobe are strongly correlated during the 20 minutes. The one on the right is the cool one. Here we're saying what changes from the first 10 to the second. And the biggest change that we saw so far with only 8 subjects is a change whereby the cross communication between the left and the right hemisphere went down in the second 10 minutes. There seemed to be a sort of a shift away from the 2 hemispheres to 1. Now, I don't want to sell you data with 8 subjects. I just want you to understand it might have some implications. Similarly, this is the Inkscape, Indira, and there were 2 major findings for Inkscape. And it's a beautiful little thing because you're doing 6 minutes of stimulation, and then you're doing all this meditation in Sanskrit, and then you do it again. And you're just looking to see what were the wisps of that experience on this simple perceptual resting state task. So the first thing is, I'm not even going to go into this. There's a lot of connectivity in general that when you're listening to, seeing those things fly and listen, there's a lot of regions, some of which are positively correlated, they're talking to one another, some that are negatively. But if you look at the change from before to after, again, we see the same pattern, which is that in the second Inkscape, there seems to be less communication between the 2 sides of the brain. What does this mean? We don't yet know. But it's potentially a marker and one that we have to dive into. Now here I'll end with the final, most interesting finding. So when you're watching the brain, there is what we call a time series. The brain activity is being charted over time. If it's an EEG, you'll see the oscillations over time. In a fMRI, you're seeing changes in the blood oxygen signal, and they're maybe going up or they're going down. But there are 2 ways you can look at this. And one of them is to look at, and this one is worth listening to me on because it turns out potentially interesting, that when we look at the brain over a time series, like the 20 minutes or the 6 minutes, you can look at the mean of the signal, like how it's changing, or you can look at whether it's highly variable. And it turns out that in each of these regions, even with a small sample, we have a reliable effect of the following. The inscape, the resting before and after, are different in that after, the signal is much more variable from region to region, from moment to moment. This is called moment to moment. And does that matter? Well, Sarah and I can talk to you about this, but there is a literature showing that sort of skilled behavior, efficient behavior, is often associated with increased moment-to-moment variation in signal, something that really changed from the resting state before to after. And we know that there is a variety of mechanisms that contribute to this. Sarah and I are now talking tomorrow about ways we would bring this into her studies and ours. But the basic idea is that something about the way the brain, whether it's moving and jumping from region to region, tends to be a good thing. This is a paper that we published in my lab years ago in the Journal of Neuroscience that was looking at kids who were better or worse readers. And we measured this trial by trial or moment by moment changes that you can see in the signal, and found that the higher that variability, the better their reading. There are a variety of measures, studies that have been done suggesting that better behavioral performance is associated with this increased variability. So what we have, and Sarah, why don't you jump in? What we have here, I'll leave that for a minute. Go ahead. What were you going to say?
I was just going to give a correlate that some of you might relate to, but you might have heard of heart rate variability. So there's also some idea that your heart is, and probably the gut too, by the way, now that I'm thinking about it, probably gut variability is going to be equally marked. Yeah. But it's well known that your heart rate also changes moment by moment based on your context, and the higher that variability is, that's associated with many health benefits. And now we're seeing this, this ability to switch of this receptive, attentive state is now becoming really an interesting literature in the brain also.
And it's a very hot topic in Sarah's field and mine. And the interesting thing is there is this evidence that there's something about the way these networks are changing moment to moment, that if it's a higher level of change, it can be associated with better performance on a variety of things. In fact, there is some indication that things like flexibility, cognitive flexibility, the ability to shift perspective, which way is that cue going? It's going this way or it's going that way. The ability to see or get a sense of a big picture is referred to as cognitive flexibility. And I'm now absorbing all these papers based on our finding just last week, showing that this variability may be associated with a very healthy ability to be flexible. One of the things that, of course, meditation is thought to affect. So I don't want to overstress this, but what I want to end with is this. The findings on especially changes in this thing called moment-to-moment variability, higher levels of which seem to be cognitively beneficial, and for which there are some published meditation studies showing this pattern. So I feel like we're on to something. And Indira, if we had enough subjects, we could look at what kind of Sanskrit experience they had on this shift in this, what's called the standard deviation of the time series versus the mean at a technical level. But it looks like we might have a biomarker that we can explore. So I want to end with 2 slides. One, the first is that it's only 8 subjects, but it happened here at the ashram, thanks to this group. And there is a lot of interesting data in this that I think we can explore with the 8 that are already there, but we also have a couple of new opportunities.
I'm just going to jump in. I know we're going to get to questions in a second. I'm going to be greedy because I don't like.
Yeah, go ahead.
But Isn't this true for all meditation, though? Don't we know that in general, that after someone meditates, there's increased, especially experienced meditators, there's going to be increased moment-to-moment variability? I mean, how novel is this compared to, or we're just excited that we found it?
No, I mean, the literature is pretty sparse. There is a bigger literature on cognitive flexibility and attentional stability,
After meditation.
and there's also some stuff on aging.
Oh, there's not much on meditation?
Not as much on meditation.
Okay.
But let me be clear. It may not be the most exciting finding in and of itself, but if it's a marker of something that should happen pre-post, then we can ask what happened in those 30-something minutes and what we manipulated. Did we give you 30 minutes of Sanskrit chanting or anything else? And to see whether it affects. So no, I'm not talking about this as entirely novel, but I'm saying that it shows us also that the data has some quality in my mind. The last thing I want to mention is that we have better system that I'm using right now in schools that has twice as many of these optodes and also gives us EEG, which gives us a very interesting look at variation in signal. And so this is the current setup compared to the one that we brought to the ashram. And I think what should strike you is we have a lot more coverage, and we also have two kinds of imaging, EEG and modality. I want to get another 15 to 20 people to do this experiment, even though this experiment wasn't perfect. And it was just a first look, to see whether we can get a deeper look at how the EEG variability, particularly the microstate changes, are affecting this, things that we're talking about. I want to end with this. So there's also another question that we can really ask ourselves, which is what's next? Now, we've done this. I think we should finish the sample, and I will, and keep in touch on this. There is a growing focus in the science of brain and behavior on designs in research that look at the same person over a long period of time. This is referred to as small n or single subject longitudinal tracking. I'm using it and my postdoc, Ahana Bhadracharya, is somebody who's done quite a lot of work with this out at Washington University. So we're now beginning to focus on watching somebody acquire mastery over a long period of time. And of course, you usually have more than one person, but you can do very exquisite science on how changes in behavior relate to changes in brain organization. So one of the things we didn't do in this first study is we didn't look at how much experience, whether you became a beginning Sanskrit or you became proficient, whether you understood the Sanskrit shlokas or you didn't, whether you could read Devanagari or not. But if we were to do this again with this nice equipment, I'm very much focused on watching small samples and tracking people over long periods of time. This is a very hot area of research development. And then I think we could come back together, the Sanskrit meditation neuroscience group, or Sarah and I talking about mindfulness in different clinical populations. We could begin to think a little bit about watching the brain evolve over an extended period. It's easy for us to put the cap on multiple times. I'm going to be in New York City this fall testing children who go through, with anxiety disorder, who are going through mindfulness training and different kinds of learning challenges to see whether a critical threshold of mindfulness will be associated with better learning. And we're going to do it with this bifurcation into the small sample. And just a reminder, when we first came here and we thought about this, Nabeen found a paper on how Sanskrit chanting changes the brain. We didn't really see anything yet because that wasn't our main goal. It was whether it changes meditation, but it would be interesting to do some studies on that. So thank you very much, and Sarah and I can answer any questions in the remaining
Let's go
time.
with RG. I've seen your hand up so long.
Go ahead.
Cap design on the addition of more electrodes, are you taking any that will actually zero in on the pineal, third eye area? since this is such a,
Repeat the question.
Yeah, Sarah, do you want to, I mean, obviously we don't cover it, but we could with MRI.
The question was whether with the high tech, with the new device, can we zero in on the third eye or the pineal gland? But Ken answer it.
Yeah, well, I was just going to say that, so with this NIRS, this cap that I brought here, right, that is limited in terms of how deep into the brain it goes, but it gives you pretty good information. EEG measures electrical activity on the scalp. It's kind of hard to know exactly where it came from, but this thing that I showed you with the eyes open and closed, fMRI, there we can localize very precisely all the way down to the brainstem. So if we had a hypothesis about the pineal or pituitary or any other site, the basal ganglia, the thalamus, all these areas that we're interested in, particularly in disease states, we can actually image those. And there are ways, I won't bore you with this, but there are ways to try to find, with many EEG channels, try to triangulate your way to specific regions. The pineal would be a tough sell for that. And certainly for the tools that we brought to the ashram, it would be off the table. But for MRI, it's possible.
We can talk more, but it's not a common area to look at for meditation research.
Exactly,
But
yeah.
Ken, why don't you?
Go,
Girija,
1 and 2, go
Girija
ahead.
and John, and then
Girija?
Indra. Okay.
I'm just like, what? Meditation creates more chitta-vritti?
Should we summarize everything a little bit, or
That's a good question.
Don't equate thoughts with the connectivity in the brain.
Exactly.
And one is the also blue was positively or negatively correlated. Can you remind me? Or
Blue is negative
negative.
between.
Okay. So the blue means that it's actually down regulation, meaning it's decrease in the connectivity. So yeah, the
variability, she's
variability.
asking. Yeah.
Oh yeah, the variability.
So this is so.
Yeah, it's it's a state. Yeah, don't confuse the thoughts, because the thought and the thinking system could regulate, but it means that the brain is kind of open, like an open slate, so that it perceives better, meaning that if there was a small noise over there, if I'm thinking, I wouldn't notice that noise. It's like you don't smell the roses, right? When you're in your own head, you don't even see what's around you. But if you're not, you are attentive. The brain can see that and hear that and feel that all at the same time. And be aware of it. It's like the brain is able to go to all different areas.
Exactly.
Just one quick example that do you know this study, by the way? But there's some showing that Buddhist monks, that if you flash like a picture on the screen of an emotion, like a crying baby, if it was up there, we could see, okay, the baby's crying, it's sad. But they gradually diminish the time, so it's so quick. But the Buddhist monks, and I'm trying to find this paper, but even the microsecond, they can sense that emotion, even though all of us would not even see the picture. Like, what? There was a picture there, but they would say, oh, it was. So they can see it, register, get meaning from it, because the brain is open to, it processes that much quicker. That's what it is.
This is a great, and Girija, I was hoping someone was going to ask with questions, so about the variance, and so let me be clear. There is a lot of science that needs to be done, is being done, on how this variation from moment to moment relates to the brain's chemistry, the glutamate, GABA, the cholinergic, things that we study, how it presents in single cells, in animals, in EEG, and in this slower thing we see with the MRI. And it's in some ways it sounds counterintuitive, but there is at least an empirical literature suggesting that more efficient states and more cognitively flexible, as Sarah says, able to shift and adapt and in this almost sort of be able to break a set in some way, seems to be associated with this signature. But to be honest, we don't really know what that signature is. And in some sense, it sounds the opposite of meditation, but it's not. But we have a long way to go, so I'm just diving into it and exploring it. And Sarah, I think, and I are both interested in it. But it's a great question.
I'm going to do a time check because it's 5 now. But how many minutes do we have? Who's the timekeeper?
I think we started.
Can we go?
It should be
Okay,
5:05.
we're gonna go. Okay, let's do the order of questions. So let's order the questions.
I think we had.
Shanta, go ahead. Who's next?
when you mention biomarkers, you mean neurotransmitters?
It could be anything. It could be what we're looking for, anything physical. So it could be the EEG signal. It could be that area of the brain. If the language is here, then that could be our biomarker. Does it go up or down? It just means what is the entity, the physical entity we're looking at, meaning, yeah.
And in the sort of cognitive neuroscience imaging world, anything that's reliably associated with behavioral change can be thought of as a biomarker. It might be structure, it might be function, but I do research with magnetic resonance spectroscopy, and we just completed a large study of children where we look at glutamate, GABA, creatine, choline, NAA. I know you're in research. And we also have this signal on every child, so we are just about to go after it. Is there a relation? Because the animal models suggest that the variance may be associated with the E to I ratio, excitatory glutamate to GABA. So there is actually, there are a variety of tools, even with human imaging, to try to see whether some of the biomarkers are essentially neurochemical. But in the jargon, anything that's reliable and is a signature of something in meditation or in anything is a biomarker. Does that? Yeah. Go ahead.
Repeat the question. The question was, what part of the data gave us confidence in that finding, that just the validity of the findings?
So you're a scientist as well. So with only 8 subjects, but with this long time series and the standard deviation of that time series, which is what we're measuring, we have significant t-tests in these 5 regions, and that's not a lot of subjects to get. So it's a pretty robust change. And again, to Girijan's point, we don't exactly know what that variance means, but it's associated with good things, with attention, and with flexibility, it appears. But that's why I'm not here to say we've discovered something new. As Sarah said, this may be out there as well. What I am saying is we brought our little toy to the ashram. We only got 8 people, but it was a great study. There's enough data in here for me to say, especially with better equipment, for me to say, I want to go further into the questions we wanted to answer, including the impact of Sanskrit on those 20 minutes and with these pre and post markers. So I'm kind of excited. But in any case, these are reliable with a relatively small n, but fairly big data set.
So is it like that you saw more activity on the right side of the brain?
It's bilateral. So these both are left, and these 3 are right. In fact, it's almost everywhere. Where we had those optodes measuring the receptors and detectors, wherever we had them, most areas were either ballpark or reliable. So it was a global effect. But the global effect is one of a real noticeable shift in signal standard deviation, not amplitude, especially with bold. You're not measuring oscillation. You're just measuring changes in the blood oxygen level. So I just think, to me, it's enough. It was enough for me this week to go, now I need to go back to my neurochemistry data and start to explore this. And Sarah and I to talk about how some of the work she does with heart rate variability and other indices of cognitive change and flexibility, how we might bring this measure in.
Muscle rigidity, by the way, muscle rigidity also is just so reactive and responsive. That's what's relevant to movement disorders. And,
I guess I would just also, I hope this is, I was called back today, Sarah and I, to simply report what we did, what I did on my summer vacation last year. And it took us forever, and we were really sorry that it took so long, but we've done a deep dive.
Can we get 2 more? Can we just get, maybe
we'll
do it really quickly.
I'm sorry, I thought
there was
we were
2
at the
more.
end.
Let's
Go ahead,
hear them
Indira.
and see if we can do it quickly. Indira first and then John.
Go ahead, John.
Indira.
Indira.
I think the variability thing is fascinating, and you explained that well. My neuroanatomy teacher in college was real big on the connections between both sides. And do you guys have any hypotheses why having less connections, and was it several, I mean, there's a bunch of different connections connecting
Yeah.
bridges. I forget what they're called
Yeah.
now. But
In the brain.
I couldn't find anything.
Yeah. I don't know. It's really interesting. I mean, the two hemispheres are connected, really. The corpus callosum, there's one main area where they are. My sense is that, you know, what resonated with me is that for the meditator studies with EEG, we see coherence from the front to the back. And I feel that it really actually does allow the brain to specialize more, so that you don't actually want the two sides actually that connected, because it allows greater, there's more of the brain to do different things at the same time. If they're bound together, that's a rigidity of the brain that limits it. So I think there's a lot of folklore in saying right brain, left brain.
Yeah,
so
this is not
not sure.
about that.
Yeah.
Yeah.
no,
Yeah,
I
but-
just
right.
I remember
Yeah, yeah.
my
Yeah,
instructor's
yeah,
bias
yeah.
was that there was power in
In
the
the-
two sides
well-
talking to each other.
well,
and
it's-
yours seem to be saying the opposite, which is really
You don't want them to be bound together. There's a difference between talking and communicating. Like there's super yoga, right, where you cross all the time. Different parts are doing different parts in the body, but you want them to be able to come in and out of sync, but also be able to break out of sync, and that's the part that's helpful. I'll just say for the Inscapes, like part of what Inscapes does, it gives you lots of beautiful visual imagery.
Right.
But at some point you might get bored and try to drift, right? But what this is showing is that person was attentive, that the samples were more attentive. So anytime a new stimuli came, a part of the brain would register it and react to it.
Right. But I would just say I'm very humble about, I think, this hemispheric effect. I'm much more bullish on this variant because it looks strong.
Right, yeah.
But
Okay.
to be honest, if you look at the meta-analyses of meditation studies, you do tend to see that the areas that are reliable in the parietal frontal
Yeah.
tend to be intrahemispheric, not cross-hemispheric.
Yeah, correct. And that's what it is. It's right in the midline. That's really kind of the connector.
Exactly.
Yeah, and so
Yeah.
actually it's really very midline that gets affected. So it's kind of that midline controller gateway. I don't know what it is.
But when we finish this sample and we also have EEG data on people, we'll just continue to do the study as we did it, I think, although we should talk about it over the next couple of days, over lunch. Like, what would we do next? And can we track people who are going from being beginning Sanskrit to experts? Because we have the equipment, we can do it. And there's methods of data analysis that are reliable with single subject, so that you can really dive deep into a person, if you will, with these measures. And that's what I'm doing in my NIH work with kids, and I think it would be interesting with the patient studies and with this here. But the hemispheric difference, let's see how that holds up. I think the other thing that was clear, and I'm confident, feel pretty good about, is we were able, using that something called independent components analysis, to identify distributed networks that hang together differently in those four different things: Inscape, Sanskrit, meditation, Inscape, Pugh. So we have a bit of a roadmap into the components of the brain that are associated with the components of the task. Now, if we have somebody who's better at Sanskrit or gets more exposure or is able to bring something different, we at least would have in fact, this is really what we get. We have now hypotheses about what to see in connectivity, what to see in activation, what to see in variability. And I think there's enough in there to say, especially with these generally advanced meditators that put the cap on here, that, it's useful. Well, we'll see.
Last question from John.
John.
This is similar to Girija and Indira. Basically, when you show the second 10 minutes of the meditation, there's less connectivity. Is that simply because there's less activity, because you think the second half, and is that correct?
I think so.
When the mind comes down, there's less activity.
I just want to repeat the question first. Yeah.
Would the variability be just a higher amplitude with a lower frequency?
No, because...
Wait, repeat the question.
Oh, sorry.
For the listening.
Go ahead, John. So John's question is first, is if you go into a more quiet state, which you clearly do, because there is that, this component that was detected is a reduction in signal over 20 minutes. So that is happening. How that affects standard deviations, though, is a little complicated because it's not an oscillation. This is a sort of a blood oxygen signal at different points in time. It could be, though, that they're correlated, variance and mean and amplitude. The research that I've just been looking at removes those effects. The paper that my lab published, what we did is we removed all of those contributors, including mean and other factors, to see if variance itself in a vacuum had an effect related to how good you were at this task, and it was.
And it's variance in oxygen level?
Yeah, in this case, this was fMRI, so it's and similar to the FNIRS, we're measuring the oxygen deoxyhemoglobin, which is slow but localized. The EEG is, I didn't get to it today, but there is a literature. In fact, there's a paper here that we were going to talk about. I'll just tell you quickly. This is a group watching people become experts over a 3-month retreat for meditation training. There is a really nice design and a lot of power, and they were measuring the fast time scales. You know, EEG is in milliseconds. The fMRI is in seconds, right? And they measured the change over in these states, these oscillatory states. And what they found, John, that I find potentially interesting is a change in something they call microstate, which just means a kind of a shift in connectivity and sort of bumps and jumps that were increasing, that were changing with the advanced meditation. So this was a pretty nice example of what you might see if you look at rapid time series versus the slower time series. The devil's in the detail on all of this. The issue is, why is this important? Well, to me, if I can get a good biomarker of the benefits of Sanskrit or meditation or reading instruction or bilingualism or anything else, then I can ask questions about individual differences. Why is it harder for this person? And this other study that we put on the board, this is actually worth a second as well. This was a study done at a group at MIT, and what they did is they took a very advanced master of the Buddhist jhana meditations. So there are 8 jhanas, going from concentration to samadhi to a sort of a non-cognitive states here. And this person went into a scanner 7 times and was able to press a button when they went into samadhi and went into these deep states. And they could reliably do it because they were a master of these 8 jhanas. And what they did is they took the MRI and they did a lot of fancy statistics, and they identified these networks that we were showing you, not the same ones necessarily, that were every time, 7 different visits, 3 hours each time. The person was going in and out of samadhi and other states, and they were able to reliably show that there were signatures of the different jhanas. Things that changed in all 8 jhanas, things that changed in jhana 3 and 6 versus 2 and 4 and blah, blah, blah. But it's fascinating. You're taking a single individual who's a master of this, and they're revealing what their brain does as they go into non-dual awareness, which is, of course, the essence of the Buddhist Sampata approach. And reliably, if their report and their physiology, which was changing, are reliable, the brain signal was extremely reliable. So I find this fascinating, and I would love to do more of this with experts.
Does that mean you could make a drug that would, if you know exactly what's happening in the brain, then you could make a drug to make samadhi?
So Girija, so this is another thing we were tempted to show, which is, this is that one I mentioned where they have people who are advanced meditators, and they're activating a brain circuit, and then what they're doing is they're stimulating the different parts. You have a little thing on, it goes zap, 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 zap.
Magnet zaps, yeah.
With magnet zaps. You can feel it, like if I zap you here, your arm will go up. Okay, it's called transcranial magnetic stimulation, or another version is TDCS, direct current stimulation. And this is being used a lot in medicine, but here's what they were proposing to do in this paper, and that is that if you look at the connections that are active in advanced meditators and you stimulate them, you can see what they're doing in that process. If somebody's struggling to meditate and you were to stimulate these, would you get a benefit? Now, nobody's done it, but it's an existence proof. Just like that single master going into the jhanas is an existence proof, this is an existence proof. So I think this is when it gets interesting to me, is when you can understand mastery, change, and plasticity, or in Sarah and I's joint work, to understand recovery of function in clinical populations. So I think we're done.
Can I ask one real quick question?
Sure.
Last question. Last question from Seth.
Last question, Seth.
And then we'll do after round. Okay, I appreciate that in the sample size of n equals 8, you saw enough to pursue this further.
I think so.
And that's wonderful. But aside from increasing the sample size to, say, 1,000, for example, you also have to control for chanting Sanskrit versus listening to Tchaikovsky, laying down
Exactly.
listening to white noise. I mean, it's very easy to say meditation is Sanskrit, but
Of course.
you really need, a study of this size is going to take an enormous amount of data and work.
No, this is what I want you to do, and I was supposed to say this. Over the next day and a half while we're here, I want you people, as we say in New York, you people, I want you people to design the next experiment that we should do. We will finish this one because it looks promising. But Sarah, Indira and Shanta, and we sat here for weeks like, well, when do we do this and why don't we do that? I think the fun of this is the equipment works, we can bring it, it's portable. What do we want to do next besides finish this study? And we had talked, for example, Sarah was just mentioning, how about 20 minutes of chanting? You know, how about different languages, as you mentioned? We do work on that as well, as you know, the bilingual representation. We can do anything we want. And really the question for meditation is Sanskrit an entry vehicle into deep meditation, and is it a kind of meditation that's different than you would get without it? And I think that's where having the chanting and then watching the 20 minutes with enough subjects, we might be able, and with some people getting no chanting, we might be able to say, this is what chanting did to your samadhi, or whatever it is we're measuring. So there's a million, but you're absolutely right. This is just like, this is almost an existence proof study, but we had enough subjects to get reliable results. Thank you, everybody.
Everybody, your questions.
Oh.
Yeah, it's okay. Thank you so much. That was exciting. It was kind of like Panini of the brain or something. There was some relationship there for sure. And can't wait to dive more into it, which we will for sure. For those outside of the room, is tonight being streamed?
Yes.
So we'll be having a wonderful Indian singer tonight. Everybody please tune in at 8 o'clock. Okay. Go to our program online if you're unsure about who's doing what. Tomorrow at 10:15 in the morning, Dave Ain Patel will be here, our wonderful Sanskrit genius, and he'll be followed by Mittal Trivedi, and then Indra in the afternoon. But thank you all, and then again on Sunday. Yeah, thank you.